Hey, Bio30s, and welcome to the second half of the Population Genetics Unit. Today I'm going to be looking or starting to look at Chapter 20 in the textbook, which is about population growth and interactions. The pages in the textbook that you'll need to be referring to for this half of the unit are going to be pages 702 to 741. Today's conversation is going to basically, focus around the population growth part of that chapter. We've talked about how we can use Hardy-Weinberg equations or the principle to determine changes to allelic frequencies in a gene pool in a population. But are there other observations we can make about populations if we're ecologists out in the wild to determine how healthy a particular population is? It isn't just about how either the gene pool is changing or staying the same, but actual changes that are perhaps quantifiable just by observation rather than having to plug them in, learning them, I guess, secondhand based on information that we're perceiving. So, for example, things like population size, population density, the distribution of the population. These are the things that ecologists are spending a lot of time looking at with populations of wild organisms, and they're using information that they're getting from those observations to determine how healthy populations are. So let's take a look at that. And as I just mentioned, the three things that we can really look at. Now, first of all, what's a population again? I've already talked about this in Bio 20, talked about it in the first half of the unit, but in case you were sleeping or didn't watch that video, what's a population? Well, a population is a group of organisms of the same species living in the same area at the same time. And what can we learn about populations just by observing them? Well, we can learn, there are three things that we can pretty much look at when we're looking at populations. We can look at their density, how tightly packed or how sparsely distributed they are per unit of area in their environment. I can look at the distribution of the population. Is there a pattern to their distribution? Is it a random distribution of the organisms? Is there maybe clumping going on where we have little pockets of the certain areas in the ecosystem that are more heavily or more densely packed with these organisms, and then other parts of the ecosystem where they're less densely packed. And then we also would look at the overall size. How many animals are living in the ecosystem, to get an idea if they're healthy or not. And sometimes too large a number could be unhealthy as well. That could be an indication of an unhealthy ecosystem. If you have, let's say, for example, a predator has been removed from an ecosystem, like we talked about wolves in Yellowstone back in Bio 20, populations of elk began to skyrocket, began to go well beyond what they should be in that ecosystem, and it began to have effects down the line as they began to eat plant material and trees, and that had a cascade effect on other organisms in the ecosystem. So whether the population is too big or too small, we want to be able to look at that population to see, what's the number and is it a healthy one. So let's take a look at some of the calculations we can do based on density and particularly size. And I will also discuss distribution of these populations in a couple seconds as well. There are four equations that are going to be coming at you today. All of them are in your data booklet, so don't panic about having to memorize them. But the first one that we'll be discussing is how do you calculate the density of a population? Well, if you've taken science before, you should pretty much know what density is, right? You're basically going to be looking at the number of organisms divided by the area that they're living in. So if I have, let's say, 1,000 bear living in an area that's 1,000 square kilometers in size, I'm going to take 1,000 bears divided by 1,000 square kilometers. That means I got one bear per kilometer living in that ecosystem. So that's a pretty standard equation. And if it's a terrestrial ecosystem, we're dealing with area. If we're in an aquatic ecosystem, the area would be measured as volume, because obviously we're dealing with volume of water rather than an actual area of space on dry land. Here's an example that's in the notes. See if you can kind of follow along at home, see how it works. We've got ecologists can estimate the density of American alligators in the Florida Everglades by counting the individuals in a few sample plots measuring 2 by 2 kilometers in size. So we've got sample plots. This is basically doing a quadrat sampling like we talked about back in Bio 20. So a 2 by 2 kilometer square, they're counting alligators. And in this case, they found 5 alligators living in that area. Calculate the average density of the alligators in this area of the Everglades. pause me if you need to, and I'm going to go over the answer here right away. Okay, well, here it is. I've got my density formula. My density of my population is going to be the number of organisms divided by the area. I do have to calculate that 2 by 2 kilometers is going to give me 4 square kilometers. There is a bit of a trick to that. So you have 4 square kilometers of area, and you've got 5 alligators living in that area. So you're going to take 5 divided by 4, and you're going to get 1.25 alligators living in every square kilometer of that section of the Everglades. So that's how you calculate density. Pretty straightforward stuff when it comes to density calculations. Let's talk about the second aspect of populations and what we can observe. What about distribution? There are, in fact, 3 major types of distribution that we'll find in nature. You've got random, uniform, and clumped distribution. Random, not very common at all. It can only occur in an environment where the environment is very uniform and the resources are equally spread out over the entire ecosystem. If there isn't an equal spread of resources for organisms in the ecosystem, they tend not to equally distribute, or I guess randomly distribute in the population, because they're going to be clumping to where the resources in fact are. An example of this would be moose in Jasper National Park would be a very good example. On the valley floors where the moose are found, there is very much a random distribution of moose. They will travel through the forest or ponds or anywhere where they have a lot of different food sources that they can have access to in Jasper National Park. So in that case, you'd be looking at an animal that has a random distribution. But generally speaking, random distributions in nature are not very common. The second type of distribution is uniform. This is where you've got a lot of what we call intra-specific competition between organisms. This would be competition between members of the same population. So territorial organisms that are fighting for space, maybe for breeding sites or for nesting sites. The example that I've got there are on the photograph are emperor penguins in the Antarctic. They tend to spread themselves out uniformly. They like to have a certain area of space around them while the males are basically nesting, are basically hatching the eggs and then keeping the chicks warm when those eggs hatch. So the analogy I would use in a classroom would be desks in a classroom, or if you want to look back to COVID, the whole social distancing. If you're a person who's not a cohort or a member of your own household, you would be trying to uniformly distance yourself from everybody else at a distance of 2 meters, right? So that would be uniform distribution. Members of your family in that uniform distribution would then clump together. So you would have certain clumping going on, and that's the most common form of distribution of a population. It's called clump distribution. This is where you've got family units that are sticking together, or they're clumping because of resources. An example of this would be like colonial dogs, like gophers or prairie dogs. The gophers on the lot beside MCHS, they've got their burrows, and even though there's lots of gophers around that field, they are going to be clumped in specific areas in the field close to their burrows for protection and for safety, right? So that is the most common form of distribution of organisms in the world, I guess, or most common you would see in nature would be the clumped distribution. Luckily, no equations related to this section. That is going to change, though, when I discuss the third and final aspect of a population, and that's the size. How do we actually calculate the size of a population? Well, one thing that ecologists do is they look to see if populations go up or go down. Is there a change in the number of organisms in a population? Because this can be a sign that if the populations are dropping dramatically, that there's a problem in the ecosystem, or there's really big pressures on that population of organisms. So how do I calculate my change in population, otherwise known as delta N? Well, in your data booklet, it's going to say something like this. It's going to say delta N is equal to factors that increase population minus factors that decrease population. They're not going to tell you what those factors are, and you are going to need to remember what those are. Well, there's only 4 factors that are going to play a role here. There's only 2 things that are going to cause a population to increase. You're either going to have births of organisms, or you're going to have organisms that actually enter the population through the process of immigration. Remember, immigration means you're going into a country or you're going into a population. That's how you remember the I, because there's also emigration, which happens if organisms leave a population. They are going to emigrate. They are going to exit the population. That's how you remember the difference between emma and emigration. So if you have The two factors that increase population are going to be births and immigration, and you're going to subtract that from the number of deaths in the population, as well as the number of organisms that actually left the population through emigration. That's going to give you your delta N of a population before and after those births and deaths occurred. So, for example, I had a population of, let's say, 100 animals, and then I had 50 births and 50 immigrations. Well, that would be an extra 100 on top of that. So that would be 100, but then I had 25 deaths and 25 emigrations. That would be 50 that I've lost, but I've had 100. I've had 100 that have entered or joined the population, 50 that have exited the population. I'm now going to have a delta N value of 50, and then my population of 100 would have increased now to 150, but my delta N value would be 50. Let's give you an example of how that works. Here's an example in the notes. There's a population of 1,641 emperor penguins on an island called Beaufort Island in the Antarctic. They're experiencing the following changes in population size between December 2015 and 2016. There are 900 births, 290 deaths, there are 245 immigrations, and there are 255 emigrations. Based on that information, calculate delta N for the population. Pause me if you need to, because here comes the answer. So basically, my delta N value is going to be equal to B plus I minus D plus E. My B value for births is going to be 900. My immigration value is going to be 245. So that's going to give me a value of 1,145. From that, I have to subtract, though, my number of deaths, which is 290, and my emigrations, which is 255. That's going to give me a total decrease number of 545. I'm going to get a delta N value of 600. When I subtract the 1145 from the 545, that's going to give me a delta N of 600. Keep in mind, if you have a delta N value that's positive, that means the population is increasing. If you have a delta N value that's negative, you know the population is decreasing. So basically, either positive or negative values is going to determine whether we have a population growth or a population that's in decline. So there's one for you. And then it also asked for, well, I didn't really ask this, but my existing population initially was 1,641. If I add to that my change in population, which is 1,600, that means that by December 2016, there should be 2,241 penguins on the island. So by using the delta N value, I can determine my new population after that 1-year study has been conducted. There you go. There's a couple of other things that we can calculate for populations as well when we're dealing with size. We can calculate two things called the growth rate and the per capita growth rate, which is known as GR or CGR, as they're abbreviated. The growth rate is basically showing the changes in the number of organisms in a population over a given amount of time. So I can calculate, okay, well, I know I've had X number of animals. So for example, the penguins we just talked about, they increased by 600 in the span of a year. So their growth rate would be 600 animals in a year. That would be their growth rate, because I would take essentially the 600 and divide it by the year, and then I could calculate their growth rate per year. I could also calculate their growth rate per month. So I could take 600 and then divide it by 12 months, and that's going to give me a growth rate of 50 penguins per month. So depending on which time value we're assigning to the study, we can calculate growth rates, but it's always based on time. Growth rate always has to do with the amount of time that's involved and how quickly a population is growing. Are they growing at a slower rate, a faster rate? That's sort of what the growth rate implies. And again, the formula for that is going to be delta N over delta T. Your N, your NF minus your NI, your number of animals, your final number of animals minus your initial number of animals, subtracted from the final time, the TF minus the TI, which is the initial time. So for example, if I gave you a study over 10 years, if I had 2020 to 2010, I could take 2020 minus 2010, that would give me 10 years. So then I know that my time factor there would be a 10-year period. And if I had, let's say, 1,000 animals increasing, if my delta N was 1,000, then it would be 1,000 divided by 10 years, would give me 100 animals per year would be the growth rate. That's how I would calculate that. Per capita growth rates are not dealing with the time. They're dealing with the number of animals that were originally in the population. For example, if I had, let's say I had 10 animals to start with, and I got 10 animals, and I got I had a growth rate of 10 animals in a year. 10 animals doesn't seem like a lot, but if my initial population was only 10, I've actually doubled my population, right? If I had a population of 1,000 and I went up by 10, it's still the same growth rate, but my per capita growth rate goes down because per animal my growth rate is not nearly as high. So I can calculate how not quickly, but how many animals for every animal that's in the population is the population going up, how fast the rate is happening based on the number of animals. And this formula is going to be delta n over n. So it's going to be my change in population divided by the initial population. So if I had a change in population, the example I just used was I had 10 animals was my delta n. My initial population was 10 animals. I would say 10 divided by 10 is going to give me essentially 1. That's going to be represented as a percentage. Now 1, if you multiply by 100, is going to give you a growth rate, a per capita growth rate of 100%. So I got a very, very big growth rate going on because it's essentially one animal for every animal that's in the population. If let's say I had 100 animals in the initial population, let's say my n value was 100, I would take 10 divided by 100 is going to give me 0.1 times 100 is only going to give me a per capita growth rate of 10%. So you get a feeling now of what the percentage value is for the growth rate in question. Let's take a look at a couple of examples to help you out here. Maybe play along at home. Hopefully you have them because there's going to be a bunch of questions coming your way after this video today. I got a population of 26 caribou that was introduced into a predator-free island in Alaska in 1910. Until 1935, the per capita growth rate was 75.9%. Calculate the number of animals living on the island by 1935. Big question. See if you can figure it out, and then I'm going to walk you through the mechanics of how we actually solve this one. Pause me if you need to, because here comes the answer right now. All right. Well, what I do have in this question is as follows. I know that, first of all, my n value for this equation is going to be 26, because I had 26 caribou to begin with. And I've also got my per capita growth rate, which is 76.9, which I'm going to convert to a decimal, which is going to be 0.769 will be my CGR. So if I rearrange the formula, delta n is going to be 0.769 times 26 caribou, which means that my increase of caribou over that 25-year period is going to be 19.7 animals. We're going to round that up to probably 20 because you can't have 0.7 of an animal. So that means I've got 20 animals that have that's my delta n value over that 25-year period. If I take 20 plus 26, it's going to give me roughly 46 caribou. That's going to be my calculation on the island by 1935. That's how I do that. Here's another question for you really quickly. In 1935, this is a follow-up question. In 1935, the food resources on the island become limited and the population crashes to 7 animals in 1937. Calculate the growth rate for that period. You are going to need to know what the animals' population was in 1935. Obviously, we know now that is 46 animals. We're going to use that as our initial population. Calculate the growth rate of that period. I'm going to let you pause the video. Calculate it if you need to. I'm going to walk you through it right now. Here's what's going to happen. My GR value is going to be delta n divided by delta t. My delta n value is going to be 7 minus 46. I have a negative growth factor because our population has dropped. My time is going to be 1937 to 1935. So I'm going to get a GR of negative 39 divided by 2, which is going to give me a growth rate of negative 19.5 caribou per year. That's how you calculate that one. All right, to finish up today, let's do a couple more things about population growth and how we can calculate them and what we can learn about organisms just by what they're doing. Each species out there has a maximum per capita growth rate. They're basically limited by how fast they can reproduce. And this is their unlimited growth rate is known as their biotic potential, which is R. If they had unlimited food, no predation, everything in life was great, this would be the maximum number of organisms that could reproduce from that species in a given amount of time. Normally speaking, this would be an exponential growth pattern if you had this happening in nature. It doesn't happen very often, but it does occasionally you would get what's called a J growth pattern. It's an exponential growth curve, population to time. It looks like a J, so we just call it a J curve. That's what it's sort of designated as in biology. This will slow down. This biotic potential is always being slowed down by things called things that are called environmental resistance. Anything in the environment that's pushing back against these animals reproducing is going to be basically considered to be environmental resistance. And that's going to push my line from this J curve to a slower growth curve. And the difference between my biotic potential and the actual curve of the population's growth in reality, the difference there in between that green area represents the environmental resistance. Well, there's two types of environmental resistance that we can deal with. There is density-dependent factors, which are biotic in nature, predation, overpopulation, anything that's related to living organisms, parasites, anything in the ecosystem that's biotic in nature. And there's also factors in the ecosystem that are abiotic, density-independent factors. These would be factors like drought, the weather's too cold. Things that are abiotic in nature, things that are not alive, that are causing pushback on the growth of the population of the organism. Eventually, most organisms, when they get out there, they're going to have an initial surge as their population grows in an ecosystem. Eventually, though, they are going to sort of kind of level out at what's called the carrying capacity. And the carrying capacity is referred to by the letter K. The carrying capacity of an environment is essentially the maximum amount of organisms that can live in that particular ecosystem based on available food and resources and stuff like that. Once we get to that stage, my J curve, which would be if there was no environmental resistance, but in most cases there is environmental resistance, my J curve becomes a sigmoidal or an S curve. So this S curve would represent a population that's sort of increasing, but then starts to slow its population increase once it hits the carrying capacity. And you can see what happens is if the population goes slightly over the carrying capacity, then the population drops off. And when it's below the carrying capacity, now it can produce a few more organisms. And it's constantly, once it gets to that carrying capacity, there's organisms that sort of hover around that carrying capacity. But there's other organisms that basically, they live hard and then they die hard. They go well past their carrying capacity, and then there's a die-off on the other side. And we'll discuss those organisms here in a couple of seconds, the differences between them. So yeah, a couple of things. Again, the rapid growing populations may overshoot their carrying capacities, as I said, causing some severe diebacks on those populations. Over time, again, the population size will fluctuate slightly around the carrying capacity. Most organisms are going to want to kind of hover around the carrying capacity because that's all the environment will allow them to reproduce to those numbers. Again, I mentioned that there were density-dependent and density-independent factors that are the environmental resistance that basically cause there to be a carrying capacity in the ecosystem. Density-dependent factors would include biotic ones, like I mentioned. These would be interspecies and intra-species competition for food territory and breeding sites, predation, parasitism, disease. These would be things that would be all density-dependent factors. Density-independent factors, again, the abiotic factors include harsh weather, temperature, natural disasters, amount of water, light, mineral nutrients in the soil. These will all play a role in what the carrying capacity is for an organism in the ecosystem. And as I mentioned earlier, the difference between the biotic potential, which is R, and the actual growth curve of the organism, the difference in the graph between them, that would represent the environmental resistance. The further over this line is, the slower the growth of the organism, and the bigger this area, the more environmental resistance is in that ecosystem, causing the slower growth rate to occur. Now, I did mention that certain organisms like to run fast and hard, and they like to reproduce very quickly, and they like to basically shoot up as quickly as they can as far as population numbers. Those organisms are called R-selected species. They are species that live close to their biotic potential. And then there's other organisms that like to have a more stable way of life. They like to basically hover their populations around the carrying capacity, or K, and for that reason, they're called K-selected species. What's the difference between them? Well, here are some of the examples of how they differ from one another. The government does expect you to know this. They could ask you a question about a species and give you several factors about a species, and you'd have to know whether they're R or a K-selected species based on what you know about them. K-selected species that live over here along the carrying capacity tend to be ones that live in stable, predictable environments. They have longer lifespans. Population sizes are stable. They have a density-dependent mortality rate, so they're more likely to be affected by biotic things in the ecosystem than abiotic. They have multiple reproductive events later in life. They take a long time to become sexually mature. They have prolonged parental care of the young, modest number of offspring, and tend to have logistical or sigmoidal growth patterns, these S growth patterns. Mammals, classic example, bigger vertebrates that tend to have longer lifespans, and mammals are kind of the textbook example of what we call a K-selected species. R-selected species, on the other hand, are pretty much the opposite. Live in rapidly changing environments. They have short lifespans. Population size is highly variable. They are affected by density-independent mortality. This would be things like temperature, water. These are the things that are going to cause these organisms to basically have pushback in their environment. They have a single reproductive event early in their life. They become sexually active very quickly. They have little or no parental care of the young. They have very high numbers of offspring, and they tend to have exponential or J-curve growth patterns. Insects, bacteria, these are examples of organisms that have, mosquitoes would be a great example of this, which are R-selected species. There you go. One last thing before I conclude. We talked about how mammals are K-selected species, and then there's humans. Yeah, humans don't play by that rule. If you look at our growth pattern here for humans, this is what humans are doing. We are growing not like K-selected species like we should be. We are growing it right now like an R-selected species. And as I mentioned, organisms that are R-selected species tend to have die-off. When they get past their carrying capacity, something's going to happen and something bad is going to happen. And just because we're humans doesn't mean that we're immune to this. We honestly don't know what the carrying capacity is for the Earth. I mean, we have technology on our side to increase that, but there's a lot of studies out there that are wondering, how many people can the Earth support before we begin to have massive die-offs of individuals because we can't feed our own species, right? So this is going to be something that we're going to have to deal with as population continues to skyrocket. All sorts of studies out there as to what populations are going to do over the next century or so. I guess you're going to be out there to find out when you get to be my age in 30 or 40 years or 50 years from now. You'll be able to see what's happening with the population. Hopefully we can continue to adapt to fulfill the needs of our very rapidly expanding population as a human species on the planet. There you go. That's it for today's lesson. Again, if you want to get a recap of everything I just discussed, watch this video here about population ecology. Great little watch there from Hank and the team at Crash Course. They always do a great job. Your homework for today, there's a booklet. Of course, there's a booklet. The booklet is entitled Population Growth Question Booklet. Lots of math in this one. So like I told you, there's going to be a lot of math in this unit. This is the last kick at the math can, though, for Bio 30. The last topic I'll be dealing with next day, a little bit more user-friendly if you're not a math head. And speaking of which, what's our conversation for next day? I'm going to finish up the course in style next day with interactions in I can't even say it. Interactions in ecological communities. We'll be discussing how communities interact with each other and all the different ways that organisms have adapted to basically are able to interact with one another and actually survive interactions with other populations in a community. That's it for today. Thanks for tuning in, and we will talk to you real soon.